fertile transgenic corn plants US 8,440,886 B2 abstract fertile transgenic zero maize corn plants which stably express heterologous DNA which is heritable are disclosed along with a process for producing said plants. The process comprises the microjectile bombardment of friable embryogenic callus from the plant to be transformed. The process may be applicable to other gramineous cereal plants which have not proven stably transformable by other techniques. Images 10 patent drawing 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 next page. Claims 12. What is claimed is 1. The fertile transgenic zero maize plant containing heterologous DNA which is heritable, wherein said heterologous DNA confers a beneficial trait to the plant, wherein said beneficial trait is selected from the group consisting of pest resistance, stress tolerance, drought resistance, disease resistance, and the ability to produce a chemically, wherein the plant expresses a selectable marker gene, and wherein the plant is from a subsequent generation of a plant that is regenerated from a selected transformed cell. 2. The plant of claim 1 wherein the heterologous DNA encodes the protein. 3. The plant of claim 2 wherein the heterologous DNA is expressed. 4. The plant of claim 1 wherein the heterologous DNA encodes an antisense RNA. 5. The plant of claim 1 wherein the zea maize plant is selected from the group consisting of field corn, popcorn, sweet corn, flint corn, and dent corn. 6. The plant of claim 1 wherein the heterologous DNA comprises a gene selected from the group consisting of non-plant genes, modified genes, synthetic genes, and genes from other plant strains or species. 7. The plant of claim 1 wherein the selectable marker gene confers resistance or tolerance to a compound selected from the group consisting of hydromycin, kanamycin, G418, neomycin, glyphosate, methotrexate, imidazolinone, chlorsulfuron, and bromoxynil. 8. The plant of claim 1 wherein the selectable marker gene confers resistance or tolerance to hygromycin. 9. The plant of claim 1 which expresses the reporter gene. 10. The seed produced by the plant of claim 1 which inherit the heterologous DNA. 11. The R2 or higher generations of the plant of claim 1, containing heterologous DNA which is heritable. 12. The plant of claim 1 which is produced from transgenic seed produced from a fertile transgenic plant using cross-breeding techniques. Description: the present application is a continuation application of U.S. Patent Application SEER number 08/677,695, filed July 10, 1996, issued as U.S. Path number 6,946,587, which is a continuation of SEER number 07/974,379, filed November 10, 1992, issued as U.S. Path number 5,538,877, which is a continuation of SEER number 07/467,983, filed January 22, 1990, now abandoned, the entire contents of which are herein incorporated by reference. Background of the invention: this invention relates to fertile transgenic plants of the species zea maize, oftentimes referred to herein as maize or corn. The invention further relates to producing transgenic plants via particle bombardment and subsequent selection techniques which have been found to produce fertile transgenic plants. Genetic engineering of plants, which entails the isolation and manipulation of genetic material usually in the form of DNA or RNA and the subsequent introduction of that genetic material into a plant or plant cells, offers considerable promise to modern agriculture and plant breeding. Increased crop food values, higher yields, feed value, reduced production costs, pest resistance, stress tolerance, drought resistance, the production of pharmaceuticals, chemicals and biological molecules as well as other beneficial traits are all potentially achievable through genetic engineering techniques. Once a gene has been identified, cloned, and engineered, it is still necessary to introduce it into a plant of interest in such a manner that the resulting plant is both fertile and capable of passing the gene on to its progeny. A variety of methods have been developed and are currently available for the transformation of various plants and plant cells with DNA. Generally these plants have been dicotyledonous, and some success has been reported with certain of the monocotyledonous cereals. However, some species have heretofore proven untransformable by any method. Thus, previous to this discovery, no technology had been developed which would permit the production of stably transformed zea-mays plants in which the transforming DNA is heritable thereof. This failure in the art is well documented in the literature and has been discussed in a number of recent reviews. Patricus, 1989, Whiting et al., 1988, Cocking et al., 1987. European Patent Publi Lens 270,356 McCabe et al. And 275,069 Arntzen et al. Describe the introduction of DNA into maize pollen followed by pollination of maize ears and formation of seeds. The plants germinated from these seeds are alleged to contain the introduced DNA, but there is no suggestion that the introduced DNA was heritable, as has been accomplished in the present invention. Only if the DNA introduced into the corn is heritable can the corn be used in breeding programs as required for successful commercialization of transgenic corn. Graves et al. 1986 claims agrobacterium-mediated transformation of zea-mays seedlings. The alleged evidence was based upon assays known to produce incorrect results. Despite extensive efforts to produce fertile transformed corn plants which transmit the transforming DNA to progeny, there have been no reported successes. Many previous failures have been based upon gene transfer to maize protoclasts, oftentimes derived from callus, liquid suspension culture cells, or other maize cells using a variety of transformation techniques. Although several of the techniques have resulted in successful transformation of corn cells, the resulting cells either could not be regenerated into corn plants or the corn plants produced were sterile. Rhodes et al. 1988 or, in some cases, it even turned out that the plants were in fact not transformed. Thus, while maize protoclasts and some other cells have previously been transformed, the resulting transferments could not be regenerated into fertile transgenic plants. On the other hand, it has been known that at least certain corn callus can be regenerated to form mature plants in a rather straightforward fashion and that the resulting plants were often fertile. However, no stable transformation of maize callus was ever achieved, i.e. there were no techniques developed which would permit a successful stable transformation of the refinable callus. An example of a maize callus transformation technique which has been tried is the use of agrobacterium-mediated transfer. The art was thus faced with a dilemma. While it was known that corn protoclast and suspension culture cells could be transformed, no techniques were available which would regenerate the transformed protoclast into a fertile plant. While it was known that corn callus could be regenerated into a fertile plant, there were no techniques known which could transform the callus, particularly while not destroying the ability of the callus both to regenerate and to form fertile plants. Recently, a new transformation technique has been created based upon the bombardment of intact cells and tissues with DNA-coated microprojectiles. The technique, disclosed in Sanford et al. 1987 as well as in ETHIO patent publication 331855 of J.C. Sanford et al. Based upon U.S. EAR number 161807, filed February 29, 1988, has been shown effective at producing transient gene expression in some plant cells and tissues including those from onion, maize. Klein et al. 1988a tobacco, rice, wheat, and soybean, and stable expression has been obtained in tobacco and soybeans. In fact, stable expression has been obtained by bombardment of suspension cultures of zea-mays black Mexican sweet. Klein et al. 1989, which cultures are, however, non-regenerable suspension culture cells, not the callus culture cells used in the process of the present invention. No protocols have been published describing the introduction of DNA by a bombardment technique into cultures of refinable maize cells of any type. No stable expression of a gene has been reported by means of bombardment of corn callus followed by regeneration of fertile plants, and no regenerable fertile corn has resulted from DNA-coated microprojectile bombardment of the suspension cultures. Thus, the art has failed to produce fertile transformed corn plants heretofore. The further stumbling block to the successful production of fertile transgenic maize plants has been in selecting those few transferments in such a manner that neither the regeneration capacity nor the fertility of the regenerated transferment are destroyed. Due to the generally low level of transferments produced by a transformation technique, the need for selection of the transferments is self-evident. However, selection generally entails the use of some toxic agent, e.g. herbicide or antibiotic, which can affect either the regenerability or the resultant plant fertility. It is thus an object of the present invention to produce fertile, stably transgenic, ZMAs plants and seeds which transmit the introduced gene to progeny. It is a further object to produce such stably transgenic plants and seeds by a particle bombardment and selection process which results in a high level of viability for a few transformed cells. It is a further object to produce fertile stably transgenic plants of other gramineous cereals besides maize. References cited: Armstrong, C.L., et al. 1985. J. Plant 164:207-214. Callis, J. et al. 1987. Genes and Develop 1:1183-1200. Chilton and Barnes 1983. Nucleoside Press 11:364-385. Chu, C.C., et al. 1975. SCI St. Peking 18:659-668. Cocking, F., et al. 1987. Science 236:1259-1260. To do it, et al. 1985. Procnatal SCI USA 82:7877873. Freeling, J.C., et al. 1976. Medici XXI:97-112. Graves, A., et al. 1986. Plant mold by all 7:4350. Green, C., et al. 1975. Crop SCI 15:417-421. Green, C.E. 1982. Plant tissue culture, a Fujiwara Education Marathon, Tokyo, Japan. Pages 107-8. Green, C., et al. 1982. Maize for Biological Research, Plant Mold by All Association. Pages 367-372. Grips, L., et al. 1983. Gene 25:179-188. Billy, H., et al. 1982. Cell 30:763-773. Jefferson, R., et al. 1987. BMBOJ 6:3009-3907. Camo, Kate, et al. 1985. Bogazet 146:327-334. Klein, T., et al. 1989. Plant Physiol 91:440-444. Klein, T., et al. 1988. Procnatal Cod SCI USA 85:4305. Klein, T., et al. 1988. The Bioflash Technology 6:559-563. Lutetium, C., et al. 1982. The Ripogen 62:109-112. McCabe, D., et al. 1988. Bioflash Technology 6:923-926. Murashig, T., et al. 1962. Physiol Plant 15:473-497. New for M. 1982. Maize for Biological Research, Plant Mold by All Association. Pages 19-30. Phillips, R., et al. 1988. Corn and Corn Improvement, Third Education, Agronomy Social A. Mer. Pages 345-387. Patrikus, I. 1989. Trends in Biotechnology 7:269-273. Rhodes, C. et al. 1988. Science 240:247. Sandbrook, J. et al. 1989. Molecular Cloning, a Laboratory Manual, Second Education, Cold Spring Harbor Laboratory Press. Sonford, J. et al. 1987. J. Part SCI and Tech 5:2737. Weising, Kate, et al. 1988. And Rev of Genetics 22:421-478. Janusz Perone, L., et al. 1985. Gene 33:109-119. Summary of the invention. The present invention relates to fertile transgenic ZMAs plants containing heterologous DNA, preferably chromosomally integrated heterologous DNA, which is heritable by progeny thereof. The invention further relates to all products derived from transgenic ZMAs plants, plant cells, plant parts, and seeds. The invention further relates to transgenic zero maize seeds stably containing heterologous DNA and progeny which inherit the heterologous DNA. The invention further relates to a process for producing fertile transgenic zero maize plants containing heterologous DNA. The process is based upon microprojectile bombardment, selection, and plant regeneration techniques. The invention further relates to a process for producing fertile transformed plants of gramineous plants other than zero maize which have not been reliably transformed by traditional methods such as electroporation, agrobacterium, injection, and previous ballistic techniques. The invention further relates to regenerated fertile mature maize plants from transformed embryogenic tissue, transgenic seeds produced therefrom, and ART1 and subsequent generations. In preferred embodiments, this invention produces the fertile transgenic plants by means of a DNA-coded microprojectile bombardment of clumps of friable embryogenic callus, followed by a controlled regimen for selection of the transformed callus lines. Brief description of the drawings. Figure 1a shows a map of plasmid vector Fig1 utilized in example I. Figure 1b shows the relevant part of Fig1 encompassing the HPT coding sequence and associated regulatory elements. The base pair numbers start from the 5 nucleotide in the recognition sequence for the indicated restriction enzymes, beginning with the Eckery site at the 5' end of the CAB35S promoter. Figure 2a shows a map of plasmid vector PBI221 utilized in example I. Figure 2b shows the relevant part of PBI221 encompassing the GUS coding sequence and associated regulatory elements. Figure 3a depicts the Phi1 containing fragments employed as probes in a southern blot and analysis of PH1 callus. Figure 3b is a southern blot of DNA isolated from the PH1 callus line and an untransformed control callus line. Figure 4a depicts the Phi1 containing fragments employed as probes in southern blot analysis of PH1RO plants. Figure 4b is a southern blot of leaf DNA isolated from RO plants regenerated from PH1 and untransformed callus. Figure 5a depicts the Phi1 containing fragments employed as probes in southern blot analysis of PH1R1 plants. Figure 5b is a southern blot of leaf DNA isolated from R1 progeny of PH1RO plants and untransformed RO plants. Figure 6a depicts the Phi1 containing fragments employed as probes in a southern blot analysis of PH2 callus. Figure 6b is a southern blot of DNA isolated from the PH2 callus line and an untransformed control callus line. Description of the preferred embodiments. The present invention is directed to the production of fertile transgenic plants and seeds of the species zero maize and to the plants, plant tissues, and seeds derived from such transgenic plants, as well as the subsequent progeny and products derived therefrom. The transgenic plants produced herein include all plants of this species, including field corn, popcorn, sweet corn, flint corn and dent corn. Transgenic is used herein to include any cell line, callus, tissue, plant part or plant which contains heterologous DNA that was introduced into plant material by a process of genetic engineering, or which was initially introduced into a plant species by such a process and was subsequently transferred to later generations by sexual or asexual cell crosses or cell divisions. By heritable is meant that the DNA is capable of transmission through a complete sexual cycle of a plant, i.e. passed from one plant through its gametes to its progeny plants in the same manner as occurs in normal corn. The transgenic plants of this invention may be produced by establishing friable embryogenic callus from the plant to be transformed, i.e. transforming said cell line by a microprojectile bombardment technique, i.e. controllably identifying or selecting transformed cells, and i.e. regenerating fertile transgenic plants from the transformed cells. Some of the plants of this invention may be produced from the transgenic seed produced from the fertile transgenic plants using conventional crossbreeding techniques to develop commercial hybrid seed containing heterologous DNA. Plant lines and tissue cultures. The cells which have been found useful to produce the fertile transgenic maize plants herein are those callus cells which are regenerable, both before and after undergoing a selection regimen as detailed further below. Generally, these cells will be derived from meristematic tissue which contains cells which have not yet terminally differentiated. Such tissue in gramineous cereals in general and in maize, in particular, comprise tissues found in juvenile leaf basal regions, immature tassels, immature embryos, and coleocular nodes. Preferably, immature embryos are used. Methods of preparing and maintaining callus from such tissue and plant types are well known in the art, and details on so doing are available in the literature. C.F. Phillips et al. 1988, the disclosure of which is hereby incorporated by reference. The specific callus used must be able to regenerate into a fertile plant. The specific regeneration capacity of particular callus is important to the success of the bombardment/selection process used herein, because during and following selection, regeneration capacity may decrease significantly. It is therefore important to start with cultures that have as high a degree of regeneration capacity as possible. Callus which is more than about 3 months and up to about 36 months of age has been found to have a sufficiently high level of regenerability and thus is currently preferred. The regenerative capacity of a particular culture may be readily determined by transferring samples thereof to regeneration medium and monitoring the formation of shoots, roots, and plantlets. The relative number of plantlets arising per petri dish or per gram fresh weight of tissue may be used as a rough quantitative estimate of regeneration capacity. Generally, a culture which will produce at least 1 plant per gram of callus tissue will be preferred. While maize callus cultures can be initiated from a number of different plant tissues, the cultures useful herein are preferably derived from the mature maize embryos, which are removed from the kernels of an ear when the embryos are about 1 to 3 mm in length. This length generally occurs about 9 to 14 days after pollination. Under aseptic conditions, the embryos are placed on conventional solid media with the embryo axis down scutellum up. Callus tissue appears from the scutellum after several days to a few weeks. After the callus has grown sufficiently, the cell proliferations from the scutellum may be evaluated for friable consistency and the presence of well-defined embryos. By friable consistency is meant that the tissue is easily dispersed without causing injury to the cells. Tissue with this morphology is then transferred to fresh media and subcultured on a routine basis about every 2 weeks. The callus initiation media is solid because callus cannot be readily initiated in liquid medium. The initiation/maintenance media is typically based on the N6 salts of Chu et al. 1975 as described in Armstrong et al. 1985 or the MS salts of Murashig et al. 1962. The basal medium is supplemented with sucrose and 2,4-dichlorophenoxyacetic acid 2,4-D. Supplements such as L-proline and casein hydrolysate have been found to improve the frequency of initiation of callus cultures, morphology, and growth. The cultures are generally maintained in the dark, though low light levels may also be used. The level of synthetic hormone 2,4-D, necessary for maintenance and propagation, should be generally about 0.3 to 3.0 mg/liters. Although successful transformation and regeneration has been accomplished herein with friable embryogenic callus, this is not meant to imply that other transformable regenerable cells, tissue, or organs cannot be employed to produce the fertile transgenic plants of this invention. The only actual requirement for the cells which are transformed is that after transformation they must be capable of regeneration of a plant containing the heterologous DNA following the particular selection or screening procedure actually used. DNA used for transformation. The heterologous DNA used for transformation herein may be circular or linear, double-stranded or single-stranded. Generally, the DNA is in the form of a plasmid and contains coding regions of beneficial heterologous DNA with flanking regulatory sequences which serve to promote the expression of the heterologous DNA present in the resultant corn plant. Heterologous DNA is used herein to include all synthetically engineered or biologically derived DNA which is introduced into a plant by man by genetic engineering, including but not limited to non-plant genes, modified genes, synthetic genes, portions of genes, as well as DNA and genes from maize and other plant